Hello everyone, so this time we are going through a paper 2, higher tier for biology. So paper 2 consists of homeostasis and response, variation inheritance, and anything to do with ecology. So there's only 3 topics on this, but they are quite chunky and there's quite a lot of content within each one that can possibly have questions on. So let's get started. There are 2 types of reproduction, sexual and asexual reproduction. Complete table 1 to compare sexual reproduction with asexual reproduction. Write a tick in the box if the statement is true. The first row has been completed for you. Now this is worth 2 marks. So there are 6 boxes to tick. In order to get 1 mark, you need 1 or 2 rows correct. So obviously these are your rows. Okay, so you need 1 or 2 of them correct. In order to get the full 2 marks, you need to get all 3 of them correct. So cell division occurs in both. Fertilization occurs. Well, fertilization requires 2 parents, so that would be sexual reproduction. Genes are passed on from parent to offspring. That would be in both of them. There's just a variation or a mixture within sexual reproduction. In asexual, they are a clone, but there's still genetic information being passed. And offspring are genetically identical to each other. Is just asexual reproduction for the most part. Obviously, in sexual reproduction, you can get clones, but they are identical twins, and that is an incredible rarity, not the common. Gametes are formed in sexual reproduction. Name the male gamete formed in flowering plants. If you have allergies, you will know this really well. It is, of course, pollen. Okay, pollen is the only answer there. Cell division by meiosis forms gametes. Figure 1 shows the mean mass of DNA per cell before, during, and after meiosis. So we've got here mean mass of DNA per cell in arbitrary units, which is here. We then have time in hours, and what we've got is mean mass is 4. Then we've got it going up to 8. I wonder what process could be happening there. Then we drop back down to 4. Oh, I wonder what process could be happening there. And then we drop down to 2. Okay, so remember that meiosis is when you get gametes. And first thing you have to kind of recollect about gametes is they contain half the genetic material of a normal body cell. So clearly what we've got here is normal body cell, just before mitosis, after mitosis, after meiosis. That's what we've got going on here. So using information from figure 1, which is this great big thing here, to answer the questions. When is the DNA in the chromosome being copied? Let's have a look. So is it between 0 and 3 hours? Got any copying there? Is it between 3 and 4? Have we got any copying there? Is it between 4 and 5, or is it between 5 and 6? Clearly, hopefully, you figure that out for yourself. It is between hours 3 and 4. So we've got this graph again we've just been talking about on the previous slide, in the previous question, well, same question. And we've got this graph again. So we've got decided that this is the normal body cell. Then we've got duplicating, we've got halving back to the original cell DNA, and then we've got halving again. So cells divide twice during meiosis, looking at that graph. Which 2 times in figure 1 show 1 cell dividing into 2 cells? So we need to tick 2 boxes because it's said 2 times, and it's highlighted 2 twice. So you need to tick 2 boxes. You're silly if you don't, right? So what we've got here is, you can actually see we have got a division at 5 hours and we've got a division at 6. So those are the 2 that we need to talk about, because like I previously said, that's your mitosis division and these are your meiosis division. What is the mean mass of DNA in arbitrary units in a sperm cell? A sperm cell is a gamete. using this graph here. So asking you to just recall a random number using the graph. So you've got 4 in the normal body cell, 4 after mitosis. Oh, what's this at the bottom there? That must be 2. And then what is the mean mass of DNA in an embryo cell? Embryo is a fertilized egg. So if there's 2 in the sperm, there must be 2 in the egg. So it will be 4 overall. Makes sense. Question 2. Earthworms live in soil. Yeah. Feed on dead and decaying plant matter. Yep. Have a soft, moist skin and exchange gases through their skin. Fabulous. All right, now the reason they can do that is because they have got a pretty small surface area to volume ratio. Large surface area to volume ratio. So they've got large surface area, very small volume. So simple diffusion is absolutely fine for them. They don't have a specialized gas exchange surface like we do. All right, so give 2 abiotic and 2 biotic factors that could affect the size of an earthworm population. Okay, so remember that 2 abiotic factors are things that are non-biological, so things to do with the weather and the environment. And 2 biotic factors are things that are living within that environment. So you could think about food, predators, etc., disease, all that kind of stuff. So abiotic factors. First thing I would go for is level of water. That's always going to be a thing to do with anything to do with animals, plants at all. It's going to be the availability of water. That's always a good one. You could talk about oxygen as well, because these guys live in the soil. Okay, the pH of the soil, the minerals and ions in the soil. Temperature is always a good one to talk about, because if it's freezing, the soil's going to be frozen, they're going to be freezing. You could talk about soil texture. There's loads of things to talk about. All right, now, biotic factors, living. All right, we're going to talk about the availability of food. Okay, now, food is a living part of the environment. It forms part of food webs and it forms part of food chains and pyramids of biomass and pyramids of number. So you have to include it in the biotic factors. Okay, food is living until obviously it's consumed, but it is living. All right, you could then talk about, like I've mentioned previously, predators, or consumers or carnivores, or whatever you want to call it, and then finally disease or pathogens or fungi or bacteria or parasites or microbes or whatever you want to call it. I don't really like the word microbe though, to be honest. It doesn't feel very scientific for me. Pathogens is a good one, as is disease. Students investigate the population of earthworms in the soil in 2 different areas because they had nothing better to do with their time. Area A is a grass lawn and Area B is a farmer's field. Okay, let's let that sit with you for a second. Grass lawn, farmer's field. Chemical X can be mixed with water and poured onto the soil. The mixture brings earthworms to the surface of the soil but does not harm the earthworms. To be fair, you could just pour water onto the soil and it would bring them up because they think it's raining. Plan an investigation using chemical X to compare the number of earthworms per metre squared in areas A and B. Now, this is a 6 marker, so because it's 6 marks, we're looking at a levelled investigation. So what that means is that level 0 is you write nothing, or it's just utter twaddle that makes no sense at all. Level 1 is the method would not lead to a valid outcome. So you've tried, but if you were to try and repeat that investigation, you'd just be going around in loops, so you just wouldn't actually get any results. B, it might not necessarily lead to a valid outcome, but you've got quite a few steps that are identified. And level 3 is clear method, logically sequenced production of a valid outcome. All right, now, first thing, because the first thing is plan investigation using chemical X, we need to talk about chemical X first off in our method. So we need to talk about same concentration and same volume of chemical X being used on both areas. Okay, so first thing that I would write is same concentration and volume of X on each field. Okay, now you may wish to say, 2 molars, 25 litres, whatever, doesn't matter. Okay, you might want to stick some numbers in there, but it's this idea of, you've mentioned the same concentration and volume. If you're asked to investigate the effect of a given chemical or reaction or whatever, you need to always state concentration and volume. Always really, really, really important, no matter what science you're in. Okay, always remember that's going to be your first step. So we've got the same concentration and volume of X on each field. I'm then going to do the same size of area sampled using a quadrat. Okay, so I'm going to use a 1 meter squared quadrat and I am going to use that to measure the areas of my field. I'm going to do this randomly using a random number generator, because at this point now it's basically turning into a sampling method. All right, so the first bit is using a chemical, but then the rest of it is just your standard, how would you measure the number of daisies in this field, how would you measure the number, the percentage coverage of grass in this field. It's still the same thing, we've just added a little bit of a twist at the start by saying that we have to add this particular chemical first. Okay, so we're going to apply X and leave for 10 mins. We're then going to collect and count worms in each area at least 5 times in each. You like that number? 5 times is the key. 5 times in each, not just 5 times overall, because that'd be 2 in one, 3 in the other. No good. At least 5 times in each. Then finally, we're going to calculate a mean and compare. That is always going to be your final spot. Okay, calculate a mean and compare. Lovely little thing. 6 marks there, and taken what? 5 minutes to write about. Nice and easy. It is important to control the concentration of glucose in the blood. Right, glucose control question. Logging into that, bringing all that to the fore, insulin, glucagon, glycogen, etc., etc. Right, fab. Figure 2 shows the concentration. Shows how the concentration of glucose in the blood of a person changed over 4 hours. So it's not said it's a diabetic person. It's just said a person. So we've got glucose concentration in moles here, millimoles, sorry. Then we've got meal being eaten, time in hours. So you can see that after the meal is eaten, we get a spike in glucose concentration and then it comes back down to normal. Okay, so that is what would be expected in a person who has normal glucose insulin control. So give one time when the concentration of the insulin in the person's blood would be high. Okay, now in that respect, you are looking at, it says use figure 2. So you're looking is in hours, all right? So when we were looking at this as examiners, we had quite a few people just kind of like plucking a time out of the air, like, oh, midday, 12 hours. It's like, no, because it's told you to use figure 2. So if you look here, you're basically looking at when the glucose starts to rise. So it's anywhere from 1.1 to 2 hours. Okay, to be fair, I'd probably pick the peak here, which is probably, what, 1.6 and a half, 1.6, something like that. So I'd probably say 1.6 hours. Just because, for me, I'm like, that's the peak of the glucose. Fine, I'll pick that. So we've got this graph again. Explain the effect of high concentration of insulin. Explain the effect a high concentration of insulin has on blood glucose concentration. Now it's an explain the effect, and it's actually, for a higher tier question, it's actually kind of giving you the things that you need to talk about. It's giving you the effect and the explanation. Now the reason they've done that here is because usually the first couple of questions on a higher paper are the last couple of questions on the foundation. So explain the effect of high concentration. Normally, if this was a higher tier only, I'd just say that, and then it'd be 3 marks, and you'd have to identify the effect in the explanation. But it's not done here, it's asked you to talk about it. So the effect is, obviously, it lowers, or it gets less, or it decreases, any of those. And the explanation is glucose taken into cells, and that's it. Glucose taken into cells, yeah. Actually 2 marks, because that's essentially what it does. That's the effect of insulin, isn't it? You could say glucose taken into cells. You could also say stored as glycogen in liver. There's a couple of ways that you could say it. But essentially, that's 1 mark. That is 2 marks. So when I've been teaching you, I cannot stress the importance of you saying glucose is moved into cells. Glucose is stored as glycogen in the liver. You have to be really careful with your explanation, your language, because that is a mark, knowing where it goes. Person A is 1.75 meters in height and has a body mass of 52 kilos. What is person A's weight category? Now, in the exam, this table that we can see here came first, and then the question came last. Sorry, it came next. I've just put it next to it here because obviously that makes more sense because it's relevant. But just be careful of that in an exam. Now, usually in an exam situation, let me just show you. Yeah, they would have it printed like this. So you'd have the graph and then you'd have the question. So you won't have to kind of flick forward and backwards. And then the next 2 questions, you don't actually need to look at this. All right, so anytime you need to actually look at something directly, it will usually be right next to the question. On the odd occasion, you might have to flick back, okay? And weirdly, that's kind of included in the difficulty of the question, the fact that you have to be able to look back and understand that there's some earlier parts of the question where information has been given to you that you do need to kind of look back at and interpret and use. So, what is person A's weight category? So you go 1.75. So we're going to say 1.75 is about here, about halfway. And they have a body mass of 52. Okay. So we're going to go up to here. We'll say 52 is about here. So we're going to go up and we're going to meet them there. So they are in the underweight category. Okay. So person A is underweight. Person B is 1.9 in height. Give the range of body mass that would put person B in the healthy weight category. So again, weirdly, this is actually kind of comes under numeracy, your ability to read and interpret a table or graph or figure. All right, with this weirdly. All right, so this is one of those. So we go to 1.9. And we go to where they have a healthy weight. So 1.9 is here. Then they hit that and we'd come down to there. And then we'd go to here and we'd come down to there. So what would we say? 68? 68 to 90, something like that. Again, because it's by eye, there is some leeway on either side. So this actually says anything in the range of 67 to 68 and in the range of 90 to 90.5. You could have had it the other way. So you could have put from 90 to 68. It's still the same thing. Usually you put your lower number first, but you're not going to get marked down for putting the numbers the other way around. Person C is obese. A doctor thinks that person C has type 2 diabetes. The doctor tests a sample of blood from person C, and table 2 shows the results. The results of the blood test, the mean results for people who do not have diabetes. So we've got cholesterol. Okay, so we've got cholesterol here, and we've got for person C, who the doctor suspects having diabetes, and somebody who doesn't, glucose, insulin. Type 2 diabetes occurs when body cells have a reduced response to insulin. So that's correct, your body is still making lots and lots and lots of insulin, but your body just isn't responding to it anymore. Give 2 ways the results of the blood test show that person C might have type 2 diabetes. Okay, well, we need to really be looking at the glucose and insulin information, because the cholesterol one has no significant bearing on our doctor's diagnosis of whether you have diabetes type 2 or not. So first thing I would say is they have a higher glucose and they have a higher insulin. That's literally it. It just says give 2 ways. It's not said use data from the table. It's not said anything like that. It's just said give 2 ways, the results support it. So the results support it because we've got more glucose and we've got more insulin. Okay, and then finally, give 2 ways that a person can reduce the chance of developing type 2 diabetes. Exercise and a low sugar, how if I can spell, sugar or carbohydrate controlled diet. Okay, just simply saying eat a healthy diet isn't good enough. You need to be specific. Okay, having a healthy balanced diet, again, no. So exercise, carbohydrate controlled, or a low sugar controlled diet. The rapid growth in human population means that more waste substances are released into the environment. The release of substances into the environment can cause pollution. Name 1 harmful substance that could cause air pollution. Straight off, CO2. Global warming, nice and easy. You could also say things like sulfur dioxide, carbon monoxide, carbon particulates, whatever. All right, but I would always go bang, CO2 straight away, global warming. Name 3 harmful substances that could cause water pollution. Do not refer to this. So we can't mention plastic, we can't mention litter. All right, what we can mention are things like fertilizers, because remember they lead to eutrophication. We can talk about sewage. We can talk about toxic chemicals from mismanaged factories or mismanaged landfill. All right, so we can say toxic chemicals. You could also say things like herbicides, fungicides, weed killers, nuclear waste. All right, there's loads of different things you could say. These are my top 3 if you're going to have a top 3. All right, these are the ones that I would talk about. Fertilizers, sewage, toxic chemicals. Fertilizers and sewage actually end up usually having the same issue, which is eutrophication, which is where we end up with those dead rivers, streams, lakes, all that kind of stuff. Describe how substances that pollute air and water can be harmful to humans and other living organisms. Right, so we are a 6 marker again. You know what that means? This means that we are leveling our answers. So 0, no relevant content. You have written again utter twaddle that means no sense or has no relevance to the question that's being asked. Now then, this one only has 2 levels. Level 1, when you get 1 to 3 marks, is facts, events or processes are identified, but simply stating their relevance, and it is not really clear that you truly understand why you're saying it. And then level 2 is scientifically relevant facts, events, processes that are identified and given in detail to form an accurate account. So when I'm looking at my mark scheme here, as an examiner, what I have is I have like coloured dots. I have like a filled in dot, and then I have an empty kind of dot, like a bullet point. The filled in one is what would be classed as like indicative content. So for example, with the air pollution, if you just said carbon dioxide is linked to global warming, yes, that would get you in the 1. Because you've not actually said how it's harmful to humans. You just said what global warming is. You've not actually said the effect of global warming. Another thing is you could say carbon monoxide combines with hemoglobin. And then if you'd have said, so less oxygen is carried by the blood, perfect. Right, so it's this idea of making sure that you're not just stating kind of 6 things, because lots of pupils, when I was marking it, did this. They just went, carbon dioxide causes global warming, acid rain, acidic gas, nitrous oxide, sulfur dioxide causes acid rain, particulates cover leaves or causes global dimming, and then they were like, right, that's it. They didn't actually think. They thought that that was the 6 things. All right, so what you need to say is CO2 causes global warming. Then we need to talk about how it is harmful. Okay, this leads to climate change, obviously, but what that means is ice caps melting. It leads to loss of habitat. Okay, so straight away as an examiner, I'm like, oh, excellent. Okay, we're probably into literally 4 marks at this point. Okay, that's how easy it is to gain or lose them. All right, so then I'm going to say SO2 causes acid rain. Okay, and then this leads to damaged leaves, so less photosynthesis. Okay, I would then say something like maybe carbon particles cause global dimming. What's the effect of that? The effect of that is less light, so less photosynthesis. Okay, now you'll also notice that I have not mentioned water yet. That's another thing, okay? If you don't mention water, you're not going to get to that level 2, even though you've written really nicely about air. Okay, so I would say something like sewage, bacteria multiply quickly, using oxygen from the water, so animals that live there cannot respire. You could also mention fertilizers causing algae to multiply, blocking sunlight, so plants can't photosynthesize, there's lack of oxygen for respiration, fish die. You could link the fertilizers to eutrophication is another thing that you could mention. Okay. Maple syrup urine disease. Yeah, not good. Okay, it's a rare inherited human condition. All right, so MSUD is usually diagnosed early in childhood and can be controlled by having a low protein diet. Figure 4 shows the inheritance of MSUD in one family. So, as usual with these pedigree analyses, you've got the squares for male and the circles for female. The filled in squares are somebody with the disease, and the empty ones are without. Now, the allele for MSUD is recessive. Now, that's important because that must mean that you need 2 copies of it in order to have the disease, which means that within this, you are going to have, within this family, because you've got people that have it, you have got carriers, and you've got sufferers, and then you've got normal, okay? So normal have none, sufferers have both, carriers have 1. So normal has no copies of the defective gene, a sufferer has 2 copies of the defective gene, and a carrier has 1 copy of the gene, but does not suffer any of the symptoms associated with it. Give 1 piece of evidence from figure 4 that shows that MSUD is a recessive condition. Well, if we have a look here, what we're going to look at is here we have a child that has the condition, and here we have a child that has the condition, but these parents don't have it. So it must be that they're a carrier. So parents without MSUD have a child with MSUD. Okay, so that comes up a lot. This kind of pedigree analysis comes up a lot, and that is the key piece of evidence that this is a recessive condition. So we've got the pedigree analysis again, and we now need to use this to answer this question here. Persons 7 and 8, which are these two here, are expecting a fourth child. Determine the probability that the child will have MSUD. You should draw a Punnett square diagram, identify the phenotype of each offspring, and use the symbols. N for not, so capital N for not having it, and lowercase n for having it. Now, because we've just said in the previous slide that in order to have a child, both parents must be carriers. So therefore, parent 7 is this, and parent 8 is this, because they both have the dominant allele for not having MSUD, but they also carry the allele for MSUD. So then we do our Punnett square. So we go capital N there, capital N, little n, and then little n, little n. Now, we've done this. We have not identified the phenotype yet. So what I would do is I would probably do something like this. Okay, n equals carrier, and then n equals sufferer. Okay, and then the probability would be 1 in 4, so you could actually write it like that, or you could write it as 25%. Either or, that's how you would do that. You do need to identify what these are. All right, it's really important. So you might want to do either circle it and put a little arrow, circle it, put a little arrow, whatever. You must identify, even if it doesn't tell you, because sometimes it won't do that. It won't tell you that, particularly on higher tier. You're going to have to know that you need to do the cross, but you always identify what the genotypes mean for the phenotype. What is a child's characteristics going to be according to these genotypes? Figure 5 shows chemical reactions involved in the normal breakdown of some types of amino acids inside the body. So you've got some amino acids and you've got enzyme 1, and enzyme 1 turns them into either toxic substance P or ammonia. Then you have enzyme 2, which turns it into harmless product, or you have the other enzyme, which is urea. So this, by the way, happens in the liver. We talked about deamination. Okay, so that's what's happening there. Okay, so this is what happens when some amino acids are digested and broken down inside body cells. All right, both of these at the end are harmless products that can be excreted. Now, a person with MSUD cannot make that enzyme there. It says they can't make enzyme 2. One of the final products shown in figure 5 is urea. Where in the human body are the reactions shown in figure 5 most likely to occur? Now, this weirdly has shown you and me, because I hadn't read this question fully, shows you the importance of reading all this first before you go on to the next question, because the question says where in the body are these reactions shown most likely to occur. Well, it's in the liver, because we've just talked about it and identified it. We've gone, oh, deamination, yeah, that happens in the liver, cool, okay. Oh, that's what the question is, Brill. All right, so we've got this table again. Remember that we said that this happens in the liver on this side, and that somebody with MSUD cannot make enzyme 2, okay. Scientists can analyse blood samples or urine samples to see if a person has MSUD. The test identifies high concentrations of toxic substance P. Explain why the blood of a person with MSUD will have a high concentration of toxic substance P. Use information. Okay, so first thing you have to say is that they have no enzyme. So cannot break down P. Okay, so the substance still being made, so the substance P is still being made because they're making enzyme 1. Okay, and then because it's really important when they said analyze blood samples or urine samples, they're saying that it so therefore the P diffuses or moves into the blood. Okay, so that just a little bit up here about the blood samples or urine samples, you have to say how it's going to get into the blood, because this is in body cells, because it says up here that it's inside the body cell. So this is happening inside the body cell. How does it get into the blood? It must diffuse in. Okay. Explain why the urine of a person with MSUD will have a high concentration of substance P. Okay, so toxic substance filtered out by kidney, because that's what your kidney does, and there's nothing wrong with the kidney in this person. Filtered out by kidney and not reabsorbed. Okay, or not all of it will be reabsorbed. Some of it might be, because obviously it still comes up on the blood test, but it's just the fact that it's not reabsorbed, so you'll pass it back out. That's the kind of thing you need to talk about when it comes to what your kidneys do. So your kidneys are there to ensure that toxic substances are filtered out and not reabsorbed. The water and ions and glucose that your body requires will be reabsorbed up to a point. Obviously, if you are diabetic, you have glucose in your urine, because your body cannot reabsorb all of that glucose that your body isn't getting into your cells. Or your kidneys can't reabsorb it all. So finally, talking about the last part of this question with a person with MSUD, why a person with MSUD must have a low protein diet. Well, clearly we need to be linking it to the fact that we've mentioned amino acids a couple of times in this. All right, so what we need to talk about is the fact that proteins are made from amino acids. So straight off, that's one mark. Okay, so low protein equals low amino acids, so less P, so less substance P, we'll call it substance P, so less substance P is made, causing less damage. Okay, so that's what we need to say there. So that's where your 3 points come from. And this is one of those that when you're looking at it as an examiner, you're like, that's a bit harsh. Okay, loads of pupils miss, because you have to say this bit here, this causing less damage, and if you don't say that, you're not going to get that final mark point. So a lot of pupils are making the link, oh, protein's made from amino acids, so low proteins mean low amino acids, so less substance P is made. Or they were forgetting this as well. They were forgetting that link with, so this just by simply stating proteins made from amino acids isn't enough. You have to link it to, why do you need a low protein diet? Because low protein means that you're going to get less amino acids in your diet, which means that less likely chance of toxic substance P being made, causing less damage because it's toxic. So we need to link it back to that toxic, which is dangerous. Toxins cause damage. Energy flows through an ecosystem and materials are recycled. Figure 6 shows the water cycle. If you do geography, you should be fist pumping now, because obviously if you do geography, anything to do with ecosystems is just a gift. Figure 6 shows the water cycle. So you've got an arrow moving away from plants through the leaves. You've got an arrow moving away from the roots into a water body. You've got X, which is a water body going into the air. Then you've got air going into clouds, and then clouds coming back down to the ground. Name process X. Evaporation. Name the process by which water is absorbed into plants. Now again, not necessarily a paper 2 topic, osmosis, but they do like to go, oh, well, you should know this from paper 1. So just be mindful that there are certain crossover points. Give 2 uses of water in plants. Again, not necessarily a paper 2 topic. So photosynthesis is one. Transpiration is the other. You could also talk about support. Do you know, like the cell becomes turgid because so it fills the cell fills a vacuole, so that you've got that cell wall because a plant doesn't have skeleton. The cow is more efficient than the grass at converting energy. Right, actually, stop myself there. All right, I've just realised what I've done. This over here came first in the exam, and then these are the questions. So we're not going to look at that. Not going to bother. I'm going to look straight over here. Figure 7 shows a flow of energy through a food chain. The numbers are in kilojoules per metre squared per year. So figure 7, we've got sunlight, 1,700,000 kilojoules. We've got growth is 21,800. Other herbivores, 3,550. So things like wild animals. Decomposers take 15,200. Only 3,050 is used, and of that 3,050, respiration, which is just giving it to the environment, is 1,025. So that respiration is used for movement, is used for maintaining body temperature. Basically, the same things that humans use energy and respiration for. 125 goes on growth, and 1,900 of it is pooped out. How very inefficient. All right. Now, the cow is more efficient than the grass at converting energy. The energy conversion of the efficiency of the cow is 4.098%. Calculate how many more times more efficient the cow is at converting energy than the grass. The equation for energy conversion efficiency is this. So we need to know the energy used for growth, the energy input, and then we're going to times it by 100, and then we're going to give our answer to 3 significant figures. Okay, so pretty easy, but for 5 marks, and that's a bit of a gift really. It's just manipulating numbers and just looking at the numbers. So let's have a look. So we need energy used for growth, which is here. So that's 21,800. We need, and that's going to be divided by energy input. Hello, 1,700,000. Okay. Then we're going to do that, and then we're going to times that by 100, and that gives us 1.2823529, blah, 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 blah. All right, so then we're going to do 4.098 divided by 1.282, because we're going to see literally how many times does this fit into that for figuring out how many more times efficient it is. And that comes out at 3.196, but again we want it to 3 significant figures, 3.20. So a cow is 3 times more efficient than converting energy than grass is. Who'd have thought? Okay, there you are. It is more energy efficient to rear cows indoors than to rear cows outdoors. Give 2 reasons why. So if you look back at that little diagram that we've just looked at, it's the major reason that it loses most of its energy is through respiration, and a lot of that energy is used to keep warm. So we're going to have less energy lost as heat, and they're going to be moving around more if they're outside, so less energy lost as movement. Because if you think about it, that's mostly what respiration is used for. It's used for maintaining body temperature. It's used for moving yourself around. So if you don't do that, then more energy is going to be used for growth. So you're going to grow quicker, which is good for the farmer because you can sell you faster. Suggest 2 possible disadvantages of rearing cows indoors. Okay, so there's quite a few. Mostly it's this increased spread of disease, because if you're all boxed up together, then you're all going to more likely spread disease. Think of school at Christmas, which is when I'm recording this, by the way, which is probably why I can hear my voice being all croaky. Because guess what? I'm ill. Most people are at this time of year. Why is that? Because we're all inside, because it's cold and it's horrible outside, which means that we're all staying inside, which means that the ability of disease to transmit just increases tenfold. This is why people are more ill in winter than they are in summer. It's because we're all inside. Okay. Another thing is you've got more stress from the animals. There's more chance of harm occurring, more chance of aggressive behaviour. You've got extra cost of heating. And lighting, okay, that's another thing. So there's loads of different things, loads of different advantages there. Increased chance of antibiotics. So this one here, there's going to be more antibiotics. I'm going to put ABs, I'm being lazy, in meat or milk, which obviously increases the chance of antibiotic resistance occurring. Scientists found a polluted pond which had a new type of blue algae in the water. The blue colour of the algae was caused by a mutation. What is a mutation? It is a change in the DNA, or base sequence, or a base, or a gene, or part of a chromosome, or a number of chromosomes, or the genetic code. Loads of different things you can say. I prefer just changing the DNA. Alright, so this is quite a long-winded question, so I'm just going to go through the information that is given to us first before we go on to looking at the questions. So, the scientists measured the number of blue algal cells in a sample of the pond water. The scientists used a special slide which has a counting grid. This is the method used. Dilute 2.5 centimetres cubed of pond water to a volume of 10 centimetres cubed with distilled water. All right, so before we go on, something that we need to make aware of is we have some kind of dilution factor going on there. All right. We're then going to place a drop of the diluted pond water onto the special slide as shown in figure 8. These special slides do exist, so they've got these little like wells in them that we can put pond water in, or any other kind of liquid sample. We're then going to place a thick cover slip over the diluted pond water to give a depth of 0.1 millimetre of pond water. Then using a microscope, we're going to count the number of algal cells in 0.2 millimetres by 0.2 millimetres squared on the counting grid. So figure 8 shows what the special slide looks like, and figure 9 shows the view of the counting grid. So it's a little bit like sampling techniques, but instead of it being daisies on a field, it is algal cells in a bit of water. So if we were looking at these when we're counting them, we'd have to make the decision about, do we count the ones that are on the lines or not, or do we just count the ones that are in it and not, do you know what I mean? So it's like this one we probably wouldn't count. This one you'd have to make a decision about whether you count or not. Is it within that 0.22 or not? These, well, that one you wouldn't count, that one you wouldn't count, etc., etc. Okay, so you have to make that decision. Now, on to the question. So I've repeated the information that we've just looked at on the previous slide here. Now, I've flipped it over because figure 9 helps us answer 7.2, and the information about the method helps us answer 7.3. So we're going to look at 7.2 first. How many algal cells are in 0.2 millimetres times 0.2 millimetres square that is in figure 9? Use the following procedure. Now, this is great that it's given us a procedure because if you remember, I was just talking a minute ago saying you should be thinking about which ones am I counting? Am I counting the ones on the edges? Am I not? Etc., etc. Now it's actually given us a method. So as I go through this method, I would be marking off the ones that I will be counting and the ones that I won't be. So it says count everything that's in the square. Yeah, obviously we'd do that. Count all the cells that are touching the left side of the lower side. So here, I'm going to be counting this one, but these here, I am not going to count because even though they're on the left and the right and the bottom side, sorry, they are not touching the grid. So I'm not counting them. And I'm not counting that either. It's also said don't count the ones that are touching the right or the top. So I'm not going to count these. I'm not going to count that because it's outside, and I'm not going to count those two. So that's the only one really that I'm going to be counting, as well as the ones that are completely within. So what I would do is I would just put a quick cross through the ones that I am not counting. Okay, and then I would maybe even highlight the ones that I am. So I'll be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So I know that I've counted them all. Then I would get my pen and I would just write 16 there. Now, 1 week later, the scientists repeated the test and counted 14 cells, brilliant, whatever, in that same counting grid. Calculate the number of algal cells in 1 millimetre cubed of undiluted pond water. Use the scientist's second count of 14 cells. So we're going to use the 14 cells and we're looking at 1 millimetre cubed. Okay, now, if you remember on the previous slide, I said that we were dealing with some kind of dilution here. Do you remember? We've got some kind of dilution factor. Now, if you look here, we're going from 2.5 to 10. So we've got a dilution. Well, it's a quarter dilution. So it's like a 4 times dilution, quarter dilution. So we're going to keep that in mind for when we are working this out. Also, we've got a depth here. And then we've got a length and we've got a width. Okay, so first thing I need to do is I need to work out the volume of my sample, which I can do because I've got a length, a width, and a depth or a height. So my volume is going to be 0.2 times 0.2 times 0.1, which gives me 0.004. Brilliant. Okay, now within that volume of liquid, I know that I have 14 cells. So what I'm going to do is I'm going to find out the number of cells that I have in 1 millimetre cubed. Okay, because this is 0.04 millimetres. So I'm going to divide 14 divided by 0.004, and that is going to give me 3,500. Brilliant. Now, at this point in time, I have got 3,500 algal cells in 1 millimetre cubed at a dilution. Okay, now in undiluted, and I need to take this number and I need to times it by 4. Okay, because that is how much it was diluted by. So I need to times the number of cells by 4, which gives me 14,000, and that's what I'm going to write down there. Okay, now I might also, just for the examiner's point of view, talk about that being the dilution factor, or that is dilution. Okay, or it's 4 times dilution, or you might want to write it up here, because as an examiner, if you write it here, okay, you can be credited for it, but it's probably better to write it in this bit here. Okay. Suggest why the scientists diluted the pond water before placing it on the special slide. Well, if you have a look at that number that we've just looked at, 14,000. Are you going to be able to count 14,000? It's just going to completely swamp the slide, all right? You're not going to be able to count it. So the reason that they dilute it is to make it easier to count. That's why we dilute it, anything to do with those. The student repeated the scientist's method, and the scientist used a thin cover slip over the diluted pond water instead of the thick one. The liquid pulled the thin cover slip down slightly. Explain how the use of the cover slip would affect the results. Right, if it's pulling it down slightly, so if we've got this thin cover slip here and it's pulling it down slightly, it's going to be going into that dip. So if it's going into that dip, we're going to end up with a smaller volume. And if we end up with a smaller volume, we're going to get a lower cell count. So it's the fact that it's pulling it down. So instead of it being thick and flat and on top, it's pulling it down into the sample. So we're going to have less volume within there, and that's going to affect our cell count. Okay, so an echidna is a mammal that lives in Australia. Figure 10 shows an echidna. Figure 11 shows how the body temperatures of the echidna varies in warm weather and in cold weather. Now remember that echidnas are mammals, but they are egg-laying mammals, monotremes. All right, so their biology is slightly different than ours. So if you have a look here, body temperature of echidna, so we've got this in cold weather, we've got these huge fluctuations, and in warm weather it stays pretty constant. Whereas with humans, over that same time period, it's pretty much constant. All right, so now we're going to look at what the questions are. Compare the variation in body temperature of the echidna in warm weather with the variation in body temperature of the human. Use data. Now, because it's asked us to use data, we must use data and we must read this accurately. Now, if you look at these graphs, each tiny square going up is equivalent to 1 degree C. So you need to make sure that you are quoting that accurately. Okay. I am allowed a tolerance of 0.5 for the echidna and 0.1 for the human, because down here, each one of these little squares is worth 0.2 degrees C. All right, so you have to make sure that you are reading it accurately. So I would say that the echidna is more variable. Okay, and it goes from, if we have a look, and this is looking in warm weather, so we're going from 27 at its lowest to 35. And the human is pretty constant, okay, and the human's pretty constant. So again, we are going from lowest is 36.2 and the highest is 37.2. Okay, nice and easy. In cold winter months, the echidna hibernates. During hibernation, the echidna's body temperature decreases to below 5 degrees, sleeps for 17 hours a day, and its rate of metabolism slows down. Explain why the decrease in body temperature is an advantage during hibernation. That's 2 marks. So first thing that I would go for is we're losing less energy, so our energy stores are going to last longer. All right, so we're losing less energy because we're not getting up and we're not moving around and our metabolism's dropped and all that kind of stuff. So body stores last longer. Makes sense. Now, there are another various things you could say. You could also say, because there is a lower temperature gradient between the cold nights and the body temperature of the echidna, you're actually going to lose less energy, because heat moves from, thinking of physics, heat moves from where there's lots of thermal energy to where there's not a lot of thermal energy. So if they're both pretty much in balance, so there's only a little bit of a difference, it's not going to be lost as quickly. So you're going to lose less energy overall. And you could also say that you are losing less, or echidnas are losing less energy for maintaining body temperature. So more energy is available for processes that are vital for life, that keep it going. So making sure that the brain and all the other organs are functioning properly. So that's another thing that you could say. So there's 3 things that you could say there. I would go straight for loses less energy, so the body stores last longer. So it keeps it going through that hibernation, which is why animals kind of eat and eat and eat and eat during the spring and summertime, and then they kind of sleep through those harsher winter months. You've got that lower temperature gradient, or you've got that less energy being transferred, so more energy is available for those vital life processes. During hibernation, the echidna wakes up several times. Each time it wakes up, it becomes active, and its body temperature increases to over 30. Why does it increase to over 30 when it's active? All right, so first thing you need to say is because when it's movement, so movement equals respiration, and respiration releases heat. That's your 2 marks. You could also say that metabolism releases heat, which increases the rate of chemical reactions. You could also say increases the rate of enzyme activity. So it's this idea of there is a respiration link. You get up, you start moving around, you're going to need more respiration because obviously your muscles are contracting. Respiration releases heat energy. Or when your metabolism is increasing because you're awake, that increases the rate of chemical reactions. So again, increases your body temperature. An echidna can dilate and constrict blood vessels in its skin. Explain how the dilation of blood vessels, so we're looking at vasodilation, and that only can help to decrease body temperature. So really common misconception is this idea of the blood vessels moving. They don't move, they just get wider. which means that the blood flow increases to the surface. They don't move. They literally go from here to here. They're not moving. My hand isn't moving. It's just getting wider. That's the same thing with what's happening with our blood vessels. So more blood flow to surface of surface of the skin. So more energy lost, which cools the blood. That's literally it. That's the same reason that vasodilation, that's no matter what, it's in a kidney, it's a human, it's a cat, it doesn't matter. Vasodilation, more blood flow to the skin, more energy is lost, cooling the blood. That's the last thing that you need to say every time you need to. An athlete trained in hot climates and the athlete lost a large volume of water each day in sweat. The athlete's energy intake each day from food was 20,000. Evaporation of 1 centimetre, kilojoules. The evaporation of 1 centimetre cubed of sweat requires 2.5 kilojoules of energy. 40% of the athlete's daily energy intake was used to evaporate sweat. Calculate the volume of sweat the athlete lost each day if 40% of his 20,000 was used to evaporate sweat. So first thing I would do is I would work out 40% of 20,000. All right, so I would do 20,000 divided by 100 and then times that by 80, which gives me 8,000. So 40% of that is 8,000. Why have I used 40%? Because it's told me. That's the daily energy intake here, 20,000, and 40% of that is used to evaporate sweat. So I've already got 8,000. Right, now, it requires 2.5 kilojoules of energy, 1 centimetre. So I'm going to divide 8,000 by 2.5, and that is going to give me 3,200 centimetres cubed. I then need to convert that. Now, if you don't convert, you are mind-bogglingly silly, because it's told you to do it in decimeters there, it's told you to do it there, and it's even told you how to do the conversion. If you didn't do it, mind-blowing stupidity. All right, so 8,000 divided by 2.5 gives us 3,200, and then I'm going to divide that by 1,000 to give me 3.2. Easy as that. Suggest why the athlete was advised to take salt tablets each day. Okay, because of sweating. If you sweat, you're going to be releasing ions that need to be replaced. So you're replacing those lost ions. All right, now I've seen pupils in the past say things like hydrate. Why would eating or consuming anything salty help you hydrate? It wouldn't. Sweat is salty. You know that sweat is salty. So if you are sweating lots and lots, like they've said here, you know, man be sweating over 3 litres here, all right? Well, I say man, I'm assuming, all right? Clearly we're going to be replacing those lost ions. That's what it's for. You know I love these experiments. Right, so students investigated the response of plant shoots to one-sided light. Figure 13 shows how the students set up 3 experiments. So you've got experiment A, the light is coming from this side here. We've got ink marks on the shoot, and then 12 hours later, oh, look at that, it's going towards, bending towards the light. In experiment B, we've got a light-proof box, and it's just going to grow straight up. And experiment C, light from all angles growing straight up. Suggest 2 control variables the students could have used in this investigation. Okay, so we are looking at what are we keeping the same. Now, there's a variety of things that you can say, all right, but in this case, I would be like, right, volume of water, temperature, species of plant, or the type of plant, same thing. Anything to do with time, carbon dioxide, light, all right, then no, that's not being kept the same, because light is obviously changing between all 3 of these experiments, so it's not the same. So it's water, temperature, type of plant. Always the same when it comes to plant investigations. Those are the 3 things, unless you are investigating temperature, are you investigating light, the type of shoot or the type of plant and the age of that plant need to be kept the same. Describe how experiment B and C acted as controls in this investigation. So experiment B shows that response is due to light. So B and C are the controls for experiment A. This is a control, this is a control. This B is proving that this response here, where you have it bending towards the light, is purely because that there is light, and it's not just, you know, it doesn't grow that way randomly. And C is that it is response to that one-sided light, okay? And that it's not response to light growing at the top or light growing at the side. It is response to that one-sided light. So that's what we need to say again. So it's saying that it is response due to one-sided light. Give two conclusions that the students could make from the ink marks on the chute in experiment A. So in experiment A, we have got those ink marks, and as you can see, towards the tip, they are getting further and further apart. Okay, you can see that on that diagram. Towards the bottom, still close together. Towards the top, they're getting further apart. Now, what it is showing is that we have an elongation at the tip, and more growth on side away from the light. Okay, that's what we're seeing here, because you can see that these sides, it is slightly bigger than the ones on the inside. Okay, so that's what we're looking at there. Then the type of response shown by the seedling in experiment A, that is phototropism. You can say positive phototropism if you wish, but phototropism is absolutely fine. Scientists made a hypothesis that light causes auxin to move from the side of the chute nearest to the light to the side furthest from the light. Describe the evidence from figure 14. Right, so let's go through this. Auxin is a plant hormone. It's made in the chute tip. Okay, scientists investigate the role of auxin in the response of chute tips to light. Grow forward seedlings in the dark for a few days. Cut the tip off each seedling. Place them on a small block of agar jelly. Place the chute tips and agar in different conditions as shown. So D and E have been kept in the dark, F and G have got one-sided light, and it shows you where the light is coming from. And then after 24 hours, measure the mass of auxin in the agar blocks. Okay, describe the evidence from figure 14, which is this here, okay, that supports the hypothesis that light causes auxins to move from the side of the chute nearest the light to the side furthest. Okay, so I am not interested in D and E. I'm not interested in these at all because these have not been kept in the light. I am only interested in F and G. So what I need to say in F, more auxin inside furthest from light. G equals same amount on each side, and that's because F, auxin can move, and in G, auxin can't move because it's got that plate glass in the middle. Look, got this piece of glass here. Okay, now that piece of glass is important because auxins can't move through glass. All right, another scientist made a different hypothesis. Light causes the breakdown of auxin. Give the evidence to show that auxin is not broken down by light. All four blocks have same mass of auxin. Okay, so 12.6 and 12.6 fine, 17.2, which is obviously quick maths, 25.2. Yeah, okay, and it's the same for the F, so it doesn't matter. It's still there. It's not like it's disappeared. Okay, it's moved. It's not disappeared. It's moved to that side. It's not gone anywhere. All right, so that's really important.